Hello everyone, myself Sweta Srivastav, working as Assistant Professor in School of Biotechnology, IFTM University, Moradabad. Today I am going to discuss one of the most interesting topic of microbiology, that is isolation of microbes, unlocking the microbial secrets. Before going to the topic, one should know what are microbes or microorganisms. These are very small living organisms that can only be visible under the microscope. They are present everywhere, that is, in the atmosphere, soil, and water. Now, if we want to observe these microorganisms and we want to perform the various studies on them, then we have to go for the isolation of microbes. Now, why to isolate the microbes? When we want to study the pure cultures for the accurate results, that is, we want to isolate a single kind of microbial culture from a mixed culture, then we go for the isolation of microbes. Secondly, if we want to identify the unknown microbes in the various environments, then also we have to perform the isolation techniques. Third, if we want to diagnose the various infectious diseases which are caused by the pathogenic microorganism, then we have to isolate these microorganisms. And lastly, if we want to develop certain targeted treatments and therapies against these infectious microorganisms, then also one has to isolate the microbes. Now, the current isolation techniques. There are various techniques that can be used for the isolation of microorganism and getting the pure culture. These are pour plate technique, spread plate technique, and streak plate technique. One of the best techniques that can be used for the isolation of the microbes is the streak plating. It is the simplest and effective technique used for colony isolation. If we want to quantify the microbes in a sample, we go for the serial dilution. It is one of the techniques that is used to increase the space, or it is used to dilute the microbial colonies so that we can reduce their numbers and can easily observe them. Another technique that is used for the isolation is by using a selective media. Selective media encourage the growth of specific microorganism while at the same time it inhibits the growth of the other microorganism. Enrichment culture technique can also be used for isolation of the microorganism. It boosts the population of the desired microorganism by the use of certain enrichment media sources. Now, what is streak plating? As I have discussed earlier, it is one of the best techniques known till now for the isolation. So, what is the prerequisite for performing the streak plate technique? For performing the streak plate technique, one should have the culture of microorganism that can be procured by performing the spread plate technique. Spreading of microorganism across the agar plate to obtain single colonies is termed as spreading. It utilizes an L-shaped loop that spreads the culture uniformly throughout the plate. From this plate, we can perform the streak plate technique, which is a 4-quadrant sticking dilute from the sample. In this technique, we first sterilize the inoculation loop, which can be visualized in the diagram shown. Here, we collect the bacteria in the loop and then we inoculate the plate. For inoculation of the plate, one has to perform the zigzag movement that can be done in a single way streak or two-way streak or multiple way. Then after the streaking process, one can go for the sterilization of the loop because once we have used the inoculation loop, it has to be sterilized every time if we utilize it for the next experiment. After the inoculation of the microbial culture, we have to grow the microbes, and that is known as incubation. Incubation means that we provide the optimized condition for the growth. Now understanding the streak plate process. The streak plate process is a unidirectional flow method that involves inoculation, that is applying the spread culture to the agar surface, then streaking, which means the zigzag movement on the plate by the diluting of the sample to separate the individual colonies, Then incubation, which allows the growth of the isolated microorganism under the specific conditions of temperature. And then lastly, we can identify and select the well-known isolated colonies. Serial dilution. As we have discussed that before performing the straight plate technique, we have to take the culture. This culture can be procured by spread plate technique, and before performing the straight plate technique, we have to go for the serial dilution. The main purpose for serial dilution is this: the serial dilution, it dilutes or it increases the distance between the microorganism. If we isolate the microorganism from any of the sources like air, water or soil, then there are so many number of microorganisms that we cannot distinguish them. In order to increase the distance between these two microbial colonies, we go for the serial dilution. That means it is a stepwise dilution of the sample which reduces the microbial concentration. And then we can use one of the dilutants which is procured from the serial dilution for the plating technique on the agar plate by the spread plate technique. Now, after incubation, these spread plates can be used for the counting. Enumerating of the colonies determines the original concentration, and then we can calculate the CFUs, that is the colony forming units, from the spread plate technique. The serial dilution is used vastly for analyzing the wastewater and for performing the food safety testing. Now, selective media can also be utilized for the isolation of the microorganism. Selective media utilizes the nutrients and the inhibitors that regulates the growth of interest of the microorganism. It enhances the growth of the desired microorganism. On the same time, it inhibits the growth of the undesired microorganism. The examples of the selective media are McConkey agar media. It selects the gram-negative bacteria, and second is the mannitol salt agar media, which selects the Staphylococcus species. Enrichment culture can also be used to boost the number of the microorganisms by providing the specific conditions which favors the growth of the microorganism of our interest. Secondly, it can overcome the competition between the other microorganisms, as well as it enhances the growth of the slow-growing microorganisms. The examples used by the enrichment culture is the nitrogen-fixing bacteria and the hydrocarbon degrading bacteria. This can be used relevantly for the bioremediation efforts. Now, when we are isolating the microorganism, how can these isolated microorganisms be utilized? These can be utilized for identifying the different pathogens and antibiotic testing by medical microbiology, as well as they can be used for studying the microbial diversity and functions. Lastly, these microorganisms which are being utilized can also be used in the industry for producing the specific enzymes, antibiotics, and biofuels, which is essentially required day-to-day life. It is also used to ensure safety and quality of the food products. It helps to identify the novel antibiotics from the soil microorganisms. Thank you very much for patience listening, as well as I think that the knowledge provided by my slides would be useful to all of you. Thank you.